this video, you will learn about the visual sensory transduction mechanism, the visual sensory pathways, and the steps of the pupillary reflex. Visual sensory transduction begins in the photoreceptors of the retina. Rods are photoreceptors that respond to a broad range of wavelengths and allow us to see in black and white, especially in dim light. Inside each rod is the photopigment rhodopsin, a G protein coupled receptor that contains a light sensitive molecule called retinal, which is derived from vitamin A. When light strikes retinal, it changes shape from a bent or cis form to a straight trans form. This shape change activates the G protein transducin. Activated transducin stimulates the enzyme phosphodiesterase, which breaks down the second messenger cyclic GMP. As cyclic GMP levels fall, the cyclic nucleotide-gated ion channels in the photoreceptor close. This causes hyperpolarization, meaning the membrane potential becomes more negative. As a result, the photoreceptor releases less neurotransmitter onto the bipolar cells. Cones function similarly, but respond to narrower ranges of wavelengths, giving us color vision. Cone opsins are similar to rhodopsin, but are tuned to specific colors: blue, green, or red. Mutations in the cone opsin genes, especially the red or green opsins on the X chromosome, can lead to color vision deficiency. After transduction, the signal passes deeper into the retina. Bipolar cells receive input from rods and cones and relay it to retinal ganglion cells. The axons of retinal ganglion cells form the optic nerve, also known as cranial nerve 2. The optic nerves travel into the cranium and reach the optic chiasm, where the medial fibers from each eye cross the midline. Past this point, the axons continue through the optic tracts toward the thalamus. The thalamus contains the lateral geniculate nucleus, where retinal ganglion cell axons synapse with thalamic neurons. These neurons project to the primary visual cortex in the occipital lobe. Here, the brain becomes consciously aware of visual information and begins processing features such as location, movement, shape, and color. From the primary visual cortex, visual information flows into two major pathways. The dorsal stream travels toward the parietal lobe and is specialized for detecting motion and spatial relationships. You can think of this dorsal stream of visual processing as the where pathway. The ventral stream travels toward the inferior temporal lobe and processes features like color and shape that help us identify objects. We can think of this ventral stream as the what pathway. The pupillary reflex is a protective mechanism that regulates the amount of light entering the eye. First, photoreceptors in the retina detect bright light and send signals through retinal circuits. Second, these signals travel along the optic nerve as the afferent pathway to reach the brain. In the midbrain, the integration center evaluates light levels. If they are too high, it activates a bilateral parasympathetic response. The fourth step involves the oculomotor nerves, along with the ciliary ganglia and ciliary nerves, which carry the parasympathetic efferent signal to the eyes. Finally, the pupillary sphincter muscles in both eyes contract, decreasing pupil diameter and reducing the amount of incoming light. While the parasympathetic division of the autonomic nervous system constricts the pupils to protect the eye from bright light, the sympathetic division dilates the pupils when light levels are low. To summarize, visual sensory transduction begins when light activates photoreceptors in the retina. The processed signals move from photoreceptors to bipolar cells and then to retinal ganglion cells, whose axons form the optic nerve. Visual information crosses at the optic chiasm, continues through the optic tracts, and reaches the lateral geniculate nucleus of the thalamus before projecting to the primary visual cortex in the occipital lobe. From there, the brain separates information into a dorsal where pathway and a ventral what pathway. Through the pupillary light reflex, the autonomic nervous system regulates the smooth muscle of the iris to maintain a stable amount of light entering the eyes. Together, these mechanisms allow us to detect light and interpret visual information.